Hello everyone, good morning. My name is Corinna Mayer. I'm a PhD student in Niklas Björkström's group, and we are located at the Center for Infectious Medicine here at the Anna Futura building at the Huddinge campus. Today we're talking about mapping dynamic landscapes of the reproductive tract with spatial proteomics. I work with human functional endometrium biopsies. The human functional endometrium is the inner lining of the uterus, which can expand during pregnancy. It undergoes morphological and functional changes to meet the requirements during a pregnancy. The functional layer of the endometrium also undergoes hormonal, metabolic changes during the menstrual cycle, where we also see a fluctuation of the proportion and the function of the immune cells. Today I'm talking about two different projects where I've been using Maximum. Both are uterine related. The first one is about the reconstitution of the uterine immune milieu after transplantation. For this project, we have been using two different cohorts. The first cohort is about 6 HLA unmatched uterus transplantation patients where we got uterine tissue after pregnancy, either the endometrial biopsy or the deciduum. And then we had a control cohort for HLA-matched hematopoietic stem cell transplantations, where we got uterine cells from the menstrual blood. In this study, we were interested in studying the uterine immune niche regeneration. And for this, we have been using flow cytometry and single-cell RNA sequencing, but also Maxima, which is a high-throughput proteomics platform. It works with cyclic staining, which means that in one cycle, you can use up to 3 antibodies that are conjugated in FITs, PE, or APC. After the staining, the machine is imaging the slide with a 20x objective and continuing with a photobleaching step where the fluorophore is getting destroyed. For this project, I've been using a 20-marker Maxima panel, mainly structural markers, but also antibodies that help us to identify immune cells and immune cell subpopulations, as well as HLA subclasses. In those experiments, we have been using 3 endometrial biopsies from uterus transplanted patients, as well as 3 biopsies from healthy controls that are in the proliferative phase of the menstrual cycle. We had 2 well-defined questions that we wanted to ask. The first one is, are the immune cells in UTX recipients derived from the donor or the recipient? And the second question is about, does the endometrium of the uterus transplanted recipients differ from healthy women? To answer the first question about the origin of the immune cells, we have been using single-cell RNA sequencing to determine the individual SNPs and have found that in the transplanted uterus, the immune cells derive from the recipient, and as a control, we also looked at the hematopoietic stem cell transplanted women, where the immune cells derive from the donor. Yeah, and this is something that we could recapitulate with our Maxima pictures. So we had 2 uterus transplanted donors where the HLA subclass was present in the recipient, like HLA-A3 and HLA-B7, and not present in the donor. We have used CD45 to visualize immune cells and then overlapped the HLA subclass that is only present in the recipient and saw that the signal is overlapping, which means that the immune cells are derived from the recipient. Next, we spatially reconstituted the uterine microenvironment after the transplantation. In the first row, you can see an endometrial biopsy from an uterus transplanted donor, and in the second row, you can see an endometrial biopsy from a healthy control. We noticed that in the uterus transplanted donor, we see all structures, for example, in blue, the glands, in orange, the vessels, and in green, immune cell aggregates. We also find immune cells in the parenchyma that are not in aggregates. When we focus on the immune cell aggregate, we noticed that most of those cells are T cells, very few are NK cells, and some are myeloid cells and B cells, which is similar to the healthy controls. We then wanted to look more into these immune cell aggregates and performed a cell segmentation. And after the cell segmentation, we gated on 4 different immune cell populations, such as CD4 positive T cells in yellow, CD8 positive T cells in green, NK cells in red, and macrophages in blue. We then continued applying a density matrix on our immune cells, which means that we put a bubble on top of our cells, and if bubbles of other cells are overlapping, this will increase the density mean of a cell. And we then split all immune cells in 2 groups, in dense areas and non-dense areas. When we looked into the different immune cell populations, we saw that the T cells had a high proportion in dense areas, whereas the NK cells and macrophages had a high proportion in non-dense areas, which was very similar to the uterus transplanted patients. In the second project, we are displaying immunometabolic dynamics of the uterine functional endometrium. During the menstrual cycle, we have a fluctuation of organic acids, lipids, and amino acids. We also see an increase in the basal body temperature just before the ovulation, and we also observe an increase in the basal metabolic rate during the secretory phase. There has been also a recent special issue from Science Advances that focus on women's health. One of those articles was about the increased heart rate, and the researchers suggested that we should build the menstrual phase into heart-brain research to improve sex-specific care. They suggest that athletes should think about their menstrual phase when they adjust their recovery time. They even go a step further, suggesting that the cycle phase shifts can change the beta blocker response. This means that we should adjust our training and our medication across the menstrual cycle. In our project, we are interested in understanding the immunometabolic shifts that might occur during the menstrual cycle. And for this, we have been collecting 8 healthy donors, where we got matched peripheral blood and endometrial biopsy at 3 different time points during the menstrual cycle. So the first time point was collected during the proliferative phase, the second time point was collected during the ovulation, and the third time point was collected during the mid-secretory phase. We have been using different methods. For example, we have been using an endometrium atlas, which is a public single-cell RNA-seq dataset to do metabolic modeling and metabolic pathway analysis. I have been using SENES on the fresh peripheral blood and the endometrial biopsy, which is a flow-based metabolic assay. Here we can compare glucose and mitochondrial dependencies per cell populations and across the menstrual cycle. We've also been using multi-MSI, which is spatial lipidomics on a tissue section. And of course, one big part of this project was also Maxima, where I've been using a 65 antibody panel. I would say 2/3 of those antibodies are needed to identify cell types and subpopulations, and 1/3 of those antibodies are targeting proteins with a metabolic function. We are comparing by population, by niche, and over time, and we are also doing spatial neighborhoods and gradients analysis. This is an overview of all the antibodies that I've been using for my final experiments. And now I thought it could be interesting for you if I talk a bit more about my analysis strategy of this pretty big data set. So I started with my analysis with cell segmentation in MaxIQ view, where each cell got an ID. And then I exported a table with the MFI of all 65 markers, also the cell center X and Y, which tells me the spatial coordination of every cell. So once we have exported this file, we convert this CSF file into an FCS file and include a couple of more columns. For example, what time point, which experiment, and which donor. And this FCS file can be imported in Flojo, which helps us to define cell populations by gating. This is just a snapshot of a simplified gating strategy that I've been following, and now I'm highlighting the 4 main structures that we find in the endometrium, such as epithelial cells, vessels, immune cells, and the fibroblasts. We then can do a spatial quality control in MaxIQ view. So basically, in MaxIQ view, we can see the raw pictures, the raw maxima pictures. Here I'm highlighting the signal of cytokeratin, which is expressed on the epithelial cells, on the glands. CD146, which is a vessel marker. In blue, you can see immune cells, and in brown, the fibroblasts. Our annotations that we have been doing in Flojo can then be imported into MaxIQ view, and here you can see the annotation, and we can already get an idea that the annotation is consistent, but we can even overlap the raw maxima pictures with our annotation, and this helps us to understand how good our gating is. We basically do this, those gatings for all of our samples to later on create one master file with all samples. So basically, we have one master file with the MFI of all 65 markers. We have columns that help us to understand which cell type is it, and the cell center X and Y for the coordination of every cell, the cell ID, as well as understanding more about the sample itself, like which time point, which experiment, and which donor. Once we have this table, we can easily pull out a lot of information. And in my next slide, I'm showing you 3 different metabolic markers that are involved in the fatty acid metabolism. So, for example, CD36 is a surface protein for fatty acid uptake. CBT1A is involved in fatty acid beta oxidation, and PLIN2 is a protein on the membrane of lipid droplets, which is basically a proxy for lipid and fatty acid storage. Here I want to show you that epithelial cells in the mid-secretory phase, here in yellow, have the highest expression of all 3 metabolic markers compared to the early proliferative phase. And this is something that we have already seen with our raw maxima pictures. Here we're looking at one donor that gave a biopsy in the early proliferative phase and in the mid-secretory phase. This can also be confirmed with single-cell RNA sequencing. So it seems that the uterine glands, the epithelial cells in the secretory phase, are very rich in nutrients, and they are actually a great source of nutrients because they produce something that is called the uterine milk, that is rich on glucose, lipids, proteins, amino acids, and electrolytes. And this is needed for the stroma cells to decerealize, and also for the invading trophoblasts. What is not yet known is if immune cells also take advantage of this uterine milk. Exactly. What we have seen with our maxima pictures is that in the proliferative phase, we have often immune cell aggregates, nicely round, and they are pretty far away from the glands. In the secretory phase, the immune cell aggregates are smaller, and they seem to be very close to the glands. So the question is now if these immune cells are also taking advantage of this uterine milk and use it as a nutrient source. So this and a lot of other questions are still yet to be explored with the maximum. And now I just want to summarize how we are using Maxima for those two projects that I've presented. So the first project had a very targeted Maxima approach with a relatively small antibody panel. We had a clear research question. It was very focused. I've only been using MaxiQ for the analysis, and we had a fast readout. For the second project, it's a lot more exploratory. We had a large antibody panel. We had at the beginning multiple research questions, and also the research questions are evolving during our analysis. We are combining software pipelines, including different softwares, and it's more time-consuming to analyze the data. Yeah, thank you all for listening to this presentation. Thanks to Niklas, who gives me the chance to work with Maxima and those great projects. Thanks to Ruth, Farron, and Casey, who are fellow Maxima users, the clinical support at the Hudinger Hospital, without whom we would not be able to work with human samples, Jesper Andersen and Trung, our collaborators from Copenhagen, who are helping us with the multi-MSI, to all donors, and to all the grants that are supporting our research, as well as to Miltenyi, in particular the technical support, who are answering all our questions and also helping us to analyze our data. Thanks.